This video contains audio content only. There are no visual elements or images accompanying the audio. If you're expecting to see video footage, please note that this is an intentional audio-only presentation. We recommend listening to this content rather than watching the screen. Thank you for your understanding and enjoy the audio. Welcome to an extraordinary exploration of human embryonic development, a journey from a single cell to a complex, living being. This article will unfold the mysteries of our beginnings, using clear, accessible language to illuminate the intricate processes that shape us all. Prepare to witness the remarkable transformation from the initial spark of life, the union of sperm and egg, to the formation of the placenta, the lifeline that nourishes and protects the developing fetus. We'll delve into the fascinating choreography of cell division, differentiation, and migration, revealing how a single cell gives rise to the multitude of specialized cells that make up our bodies. We'll uncover the secrets of gastrulation, the process that lays the foundation for our body plan, establishing the head-to-tail axis and the three primary germ layers, ectoderm, mesoderm, and endoderm, from which all our tissues and organs arise. We'll explore the intricate processes of neurulation, the formation of the nervous system, and organogenesis, the emergence of our organs, from the beating heart to the developing lungs. This journey will not only reveal the biological marvels of embryonic development but also explore the profound ethical considerations that arise from our growing understanding of these processes. We'll delve into the complex questions surrounding the moral status of the embryo and the implications for research, highlighting the importance of ongoing ethical discussions and public engagement. By the end of this article, you will have a deeper appreciation for the intricate dance of life, the remarkable journey from a single cell to a complex human being. You'll gain insights into the delicate interplay of genes and environment, the forces that shape our individual destinies. You'll also understand the ethical complexities surrounding embryonic research and its potential to unlock new treatments and cures for a wide range of diseases. Join us on this extraordinary adventure, as we unravel the mysteries of human embryonic development and explore the profound questions they raise about the very nature of life itself. Chapter 1. The Spark of Life, from Gamete to Zygote. The human story begins, not with a cry, but with a whisper, an almost unimaginable meeting of two minuscule cells, the sperm and the egg, the fundamental units of life we call gametes. This encounter, the very foundation of our existence, is far from a simple rendezvous. It's an epic journey fraught with challenges, a biological odyssey of microscopic proportions. The saga begins with the deposit of millions of sperm, tiny vessels carrying the father's genetic legacy, into the vast expanse of the vagina. Immediately, they face a hostile environment, the first of many hurdles in their quest. The sheer physical stress of ejaculation propels them into a world vastly different from their origin. Uterine contractions, stimulated by prostaglandins present in the semen and oxytocin released during the act of coitus, create a churning current, aiding their initial transport. This involuntary muscular action, while assisting their progress, also adds to the physical challenges they face. Passively aspirated into the uterine cavity along with secretions from the uterine glands, the sperm begin their active pursuit. It's important to note that while uterine contractions and fluid currents play a role in their initial movement, the sperm's own motility, their intrinsic ability to swim, is paramount for their journey from the uterine cavity to the waiting egg. This journey is a perilous one. The cervix and uterine ostium, narrow passages within the female reproductive tract, act as formidable barriers, drastically reducing their numbers. Of the hundreds of millions initially embarked on this voyage, only about 1% manage to enter the uterus. Those that do face a daunting gauntlet of further obstacles. Shifts in pH, the acidity or alkalinity of their surroundings, threaten their delicate biochemistry. Thick mucus secretions create a viscous labyrinth, hindering their progress. And ever present, the vigilant sentinels of the female immune system stand ready to identify and eliminate these foreign invaders. Within 24 hours, the vast majority of the sperm succumb to these harsh conditions, their journey cut short. For the survivors, the journey from cervix to oviduct can take anywhere from 30 minutes to 6 grueling days. Meanwhile, a solitary matured oocyte, the egg, embarks on its own, shorter but equally crucial journey. Released from the ovary into the delicate embrace of the fallopian fimbriae, it begins its passage through the fallopian tube. Ciliary beats, rhythmic movements of tiny hair-like projections lining the tube, gently propel the egg forward. This movement is aided by the rhythmical contractions of the fallopian tube's musculature, creating a gentle current that guides the egg toward its destiny. The egg, unlike the sperm, is not built for speed. Its journey is one of careful orchestration, ensuring its arrival at the ampulla, the usual site of fertilization, at the precise time for the potential encounter. As the sperm navigate the fallopian tube, they undergo a critical transformation, a process called capacitation. This is not merely a matter of physical endurance. It's a profound physiological and biochemical metamorphosis, preparing them for the ultimate challenge. Their membranes become more fluid, allowing for the release of enzymes crucial for penetrating the egg's protective layers. Internally, their biochemistry shifts, priming them for the acrosomal reaction, a crucial step for fertilization. Most importantly, capacitation enables the sperm to respond to the siren call of the egg. The egg, surrounded by its cumulus cells, releases a bouquet of chemical signals into the fallopian fluid. Progesterone, a key hormone in the reproductive cycle, acts as a beacon, guiding the capacitated sperm towards their target. Other nonpeptide signaling molecules, smaller chemical messengers, add to this complex symphony, creating a chemical gradient that only capacitated sperm can detect and follow. This intricate interplay of chemical signals ensures that only the most prepared sperm are drawn to the egg. Of the few thousand sperm that reach the vicinity of the egg, only a select few breach the outer layer of cumulus cells. Now, they face the egg's formidable defenses, three distinct barriers: the corona radiata, a loose layer of cells; the zona pellucida, a thick glycoprotein matrix; and finally, the vitelline membrane, the egg's own cell membrane. The remaining sperm, those that don't fertilize the egg, are not mere bystanders. They contribute to the weakening of the corona radiata, and their combined efforts are crucial for the next stage, the acrosomal reaction. As the sperm approach the zona pellucida, their acrosomes, cap-like structures at the head of each sperm, release a potent cocktail of enzymes. Hyaluronidase, one of these enzymes, further disperses the cells of the corona radiata. Other acrosomal enzymes, such as acrosin, begin to digest the zona pellucida, creating a path for a single sperm to reach the vitelline membrane. Finally, one sperm, having successfully navigated this chemical and physical labyrinth, reaches the vitelline membrane. The membranes of the sperm and egg fuse, a moment of profound connection, initiating a cascade of events that will seal the egg's fate. The sperm's nucleus, carrying its precious cargo of paternal DNA, enters the egg's cytoplasm. This fusion triggers a rapid response, the cortical reaction. Cortical granules, small vesicles located just beneath the egg's membrane, release their contents into the space between the vitelline membrane and the zona pellucida. These enzymes modify the zona pellucida, making it impenetrable to other sperm, a crucial defense against polyspermy, the fertilization of an egg by multiple sperm, which would result in an unviable embryo. Inside the egg, the sperm head undergoes a dramatic transformation. It swells, its tightly packed DNA decondensing, forming the male pronucleus. Simultaneously, the egg's nucleus reassembles, becoming the female pronucleus. Each pronucleus, a sphere containing a haploid set of 23 chromosomes, represents half of the genetic blueprint needed to create a new individual. The male and female pronuclei, drawn together by an intricate dance of cellular machinery, approach each other at the center of the egg. Their membranes dissolve, and the chromosomes, the very essence of heredity, combine. This union, the fusion of the pronuclei, marks the creation of the zygote, a single cell containing a complete diploid set of 46 chromosomes, a unique blend of genetic information from both parents. This newly formed genome, the zygote's DNA, is the blueprint for the future individual, the starting point of a unique human life. The spark of life, ignited in this microscopic fusion, has set in motion a developmental journey of astonishing complexity and wonder. Chapter 2. The Dance of Creation, Implantation and the Emergence of Form. The zygote, this newly formed single cell, a microscopic sphere pulsating with the potential of a human life, doesn't linger in the ampulla of the fallopian tube, the site of its creation. It embarks on a journey, a slow, deliberate migration towards the uterine cavity, the destined cradle for its development. This voyage, lasting approximately 4 days, is not merely a passive drift. It's a period of intense activity, the beginning of a remarkable transformation driven by the intrinsic program encoded within the zygote's DNA. The first act of this developmental drama is cleavage, a process of rapid meiotic cell divisions. Within the protective embrace of the zona pellucida, the zygote divides, not by increasing in size, but by partitioning itself into smaller and smaller cells, the blastomeres. Approximately 24 hours after fertilization, the first cleavage occurs, splitting the zygote into 2 identical cells. This initial division, along the meridional axis, marks the beginning of multicellularity. Another 24 hours pass, and each of these 2 cells divides again, resulting in a 4-cell stage. The divisions continue, asynchronously now, some cells dividing faster than others, resulting in a somewhat uneven progression through a 6, 8, 10, and 12-cell stage. By the 4th day, the conceptus reaches a 16-cell stage, a cluster of cells resembling a mulberry, and thus aptly named the morula. This rapid cellular proliferation doesn't increase the overall size of the conceptus. Confined within the zona pellucida, the blastomeres become increasingly compacted, their membranes tightly adhered to one another. This compaction, far from a simple physical squeezing, is a critical event. It initiates the first differentiation within the embryo, setting the stage for the emergence of distinct cell lineages. The outer cells of the morula, destined to become the trophoblast, develop tight junctions, forming a continuous epithelial layer. These cells, in close contact with the uterine environment, will play a crucial role in implantation and the formation of the placenta. The inner cells, on the other hand, maintain a more rounded shape and develop gap junctions, facilitating intercellular communication. These cells, the inner cell mass, will give rise to the embryo proper. As the morula enters the uterine cavity, it begins to draw fluid from its surroundings, accumulating it within the intercellular spaces. This fluid accumulation, driven by the active pumping of sodium ions by the outer cells of the morula, creates an osmotic gradient, drawing water into the interior. This marks the beginning of blastocyst formation. The fluid coalesces, forming a cavity within the morula, the blastocele. The morula, now transformed into a blastocyst, resembles a hollow sphere, with the inner cell mass clustered at one pole, the embryonic pole, and the blastocele occupying the remaining space. The blastocyst, still encased within the zona pellucida, continues to expand due to the increasing fluid pressure within the blastocele. This expansion thins the zona pellucida, eventually leading to its rupture. This hatching process, occurring around day 5 or 6 after fertilization, is a critical prerequisite for implantation. Freed from the confines of the zona pellucida, the blastocyst can now interact directly with the uterine lining. The blastocyst doesn't implant randomly. It seeks a receptive endometrium, the inner lining of the uterus, which has been meticulously prepared for this encounter. This preparation, called decidualization, is orchestrated by a complex interplay of hormones, primarily estrogen and progesterone, secreted by the corpus luteum in the ovary. Estrogen and progesterone stimulate the endometrial glands to grow and secrete, enriching the uterine lining with nutrients. The spiral arteries, supplying blood to the endometrium, become coiled and engorged, increasing blood flow and providing a rich source of oxygen and nutrients for the developing embryo. The endometrial stroma, the connective tissue beneath the epithelium, becomes edematous and filled with large, glycogen-rich decidual cells, creating a succulent, nourishing environment for the incoming blastocyst. The first contact between the blastocyst and the endometrium is a delicate dance of recognition and adhesion, called apposition. The blastocyst, guided by cell adhesion factors expressed on its surface, orients itself with the embryonic pole, the site of the inner cell mass, facing the uterine lining. Polarity signaling, a complex interplay of molecular cues, ensures this precise orientation. The trophoblast cells, now in direct contact with the endometrial epithelium, begin to differentiate further. Some trophoblast cells fuse to form the syncytiotrophoblast, a multinucleated mass that invades the uterine lining, anchoring the blastocyst and initiating the formation of the placenta. Other trophoblast cells remain distinct, forming the cytotrophoblast, a proliferative layer that replenishes the syncytiotrophoblast and contributes to the developing placenta. The blastocyst, now firmly embedded in the uterine lining, transitions from a simple, hollow sphere to a multi-layered structure. The inner cell mass, nestled between the blastocele and the developing amniotic cavity, differentiates into two distinct layers, the epiblast and the hypoblast. The epiblast, the upper layer, will give rise to the three primary germ layers, ectoderm, mesoderm, and endoderm, the building blocks of the entire fetal body. The hypoblast, the lower layer, will contribute to the yolk sac, an extraembryonic membrane that plays a vital role in early embryonic nutrition and blood cell formation. This bilaminar disc, formed by the epiblast and hypoblast, marks the end of the implantation process and sets the stage for gastrulation, the next major transformation in the embryo's journey towards complexity. The dance of creation continues, each step a precisely choreographed movement in the intricate ballet of human development. Chapter 3. The Architect Within, Gastrulation and the Blueprint of Humanity. The embryo, nestled within the welcoming embrace of the uterine lining, has just completed its implantation, a crucial step in its developmental journey. But the real marvel, the true dance of creation, is about to begin. This is gastrulation, a process of dramatic transformation, a period of intense cellular choreography where the simple, two-layered embryonic disc is sculpted into a three-layered masterpiece, the blueprint for the entire human body. Imagine a flat, circular field of cells, the epiblast. Suddenly, a faint line appears at one edge, a subtle groove that deepens and extends across the surface, like a miniature version of the Grand Canyon. This is the primitive streak, a transient but incredibly important structure. It's the embryo's GPS, establishing the head-to-tail axis, the anterior-posterior direction, and the left-right symmetry that will define the body's organization. The formation of the primitive streak is not random. It's orchestrated by a complex interplay of signaling molecules. Cells at the posterior edge of the epiblast release signaling proteins, like BMP4 in primates, creating a concentration gradient that guides the formation of the streak. These signals act like chemical messengers, instructing the epiblast cells to change their shape and behavior, initiating the formation of the groove. The primitive streak is not just a marker of the embryo's axes. It's a dynamic structure, a conduit for cell migration, a gateway to a new level of organization. Cells of the epiblast, initially arranged in a simple sheet, begin to move towards the primitive streak, drawn by chemical signals emanating from the groove. This coordinated movement of cells is akin to a river converging on a central channel, a cellular flow directed by the guiding influence of the primitive streak. As the epiblast cells approach the primitive streak, their movement becomes more dramatic. They don't simply slide across the surface. They change shape, becoming more elongated and bottle-shaped. They converge on the primitive streak, jostling for position like eager spectators at a grand event. Then, one by one, they dive into the groove, a process called invagination, disappearing from the surface view and embarking on a transformative journey. Once inside the primitive streak, the cells continue their migration, spreading out between the epiblast and the hypoblast, the lower layer of the embryonic disc. This spreading movement, like a wave washing over a beach, is a form of cell movement known as involution. These migrating cells, pioneers of a new order, are destined to become the mesoderm, the middle layer of the three primary germ layers. Some of the invaginating cells don't stop at the mesoderm. They continue their downward journey, displacing the hypoblast cells and forming a new layer, the endoderm, the innermost layer of the three germ layers. The remaining epiblast cells, those that didn't migrate through the primitive streak, now constitute the ectoderm, the outermost layer. The three germ layers, formed during the intricate ballet of gastrulation, are not just simple sheets of cells. They are the progenitors of all the diverse tissues and organs that make up the human body. Their fates are not predetermined by their position, but by a complex interplay of molecular signals and genetic programs, a symphony of interactions that orchestrates the emergence of specialized cell types. The ectoderm, the outermost layer, gives rise to the body's protective covering, the skin. But it's not just a simple barrier. It differentiates into a variety of specialized cells, including keratinocytes, the tough, waterproof cells of the epidermis, melanocytes, the pigment-producing cells that give our skin its color, and Langerhans cells, immune cells that guard against infection. The ectoderm also forms the nervous system, the body's control center, differentiating into neurons, the information processing cells of the brain and spinal cord, and glial cells, the support cells that nourish and protect neurons. Sensory organs, like the eyes and ears, also arise from the ectoderm, allowing us to perceive the world around us. The mesoderm, the middle layer, is the source of the body's structural framework, the muscles and bones that enable movement and support. But it's not just about structure. The mesoderm also forms the circulatory system, the heart and blood vessels that transport oxygen and nutrients throughout the body. The excretory system, the kidneys and bladder that filter waste products from the blood, and the reproductive system, the gonads and associated structures that enable procreation. The mesoderm's diversity is reflected in the wide range of cell types it generates, from the contractile cells of the heart and skeletal muscles to the osteocytes that form bone and the chondrocytes that create cartilage. The endoderm, the innermost layer, forms the lining of the digestive tract, a continuous tube from mouth to anus, responsible for breaking down food and absorbing nutrients. It also gives rise to the lining of the respiratory tract, the lungs and airways that enable gas exchange. Organs like the liver and pancreas, crucial for metabolism and digestion, also arise from the endoderm. The endoderm cells differentiate into a variety of specialized epithelial cells, lining the inner surfaces of the body and performing a range of functions, from secreting digestive enzymes to absorbing nutrients and exchanging gases. The intricate movements and transformations of gastrulation are not random. They are guided by a complex interplay of molecular signals, a language of cells that orchestrates the emergence of form and function. Signaling molecules, like BMPs, WNTs, FGFs, and nodal, act like chemical messengers, diffusing through the embryo and influencing the behavior of their target cells. These signals activate specific genetic pathways, triggering changes in gene expression that control cell movement, division, and differentiation. Imagine a construction site where different teams of workers are building different parts of a building. Signaling molecules are like the foreman's instructions, telling each team what to do and when to do it. Some signals act as activators, like a green light, telling cells to move or divide. Others act as inhibitors, like a red light, stopping cells from moving or differentiating prematurely. The precise timing and location of these signals are crucial for ensuring that the different parts of the embryo develop in the correct sequence and spatial arrangement. Gastrulation, this intricate process of cellular choreography, is not just a biological marvel. It's a critical event with profound clinical significance. Errors during gastrulation, disruptions in the molecular signals or cellular movements, can have devastating consequences, leading to a range of developmental defects. For example, defects in the formation or closure of the neural tube, a structure derived from the ectoderm, can lead to neural tube defects, like spina bifida or anencephaly. Disruptions in mesoderm migration can result in heart defects, skeletal abnormalities, or urogenital malformations. Problems with endoderm formation can lead to defects in the digestive or respiratory systems. Gastrulation is a delicate and precisely orchestrated process, a critical period in human development. Understanding the molecular mechanisms and cellular movements involved in this process is not just a matter of scientific curiosity. It's essential for understanding the origins of birth defects and for developing strategies to prevent them. Chapter 4. The Enigma of Early Fate, Cell Differentiation and the Path Not Taken. Gastrulation, the remarkable transformation of the two-layered embryonic disc into a three-layered structure, sets the stage for an even more profound process, cell differentiation. This is where the true magic of development unfolds, the point at which the generic cells of the embryo begin to specialize, embarking on distinct paths to become the diverse array of cell types that comprise the human body: skin cells, nerve cells, muscle cells, blood cells, and the myriad other specialized components that make us who we are. This specialization isn't a random process, it's a meticulously orchestrated symphony of signals, a complex interplay of molecular cues and genetic programs. These signals, both internal and external to the cell, act as switches, turning genes on and off in specific patterns, guiding cells down particular developmental pathways. Internal signals, intrinsic to the cell itself, arise from the cell's own DNA, its unique genetic blueprint. External signals, on the other hand, come from the cell's surroundings, its microenvironment within the developing embryo. These external signals can be secreted proteins, like morphogens, that diffuse through the embryo, creating concentration gradients that influence cell fate. They can also be physical cues, like cell-cell contacts or mechanical forces, that alter the cell's shape and behavior. One example of the interplay of signals is seen in trophoblast differentiation during implantation. The outer cells of the blastocyst, upon contact with the uterine lining, are exposed to signals, including BMP4, that trigger their differentiation into the syncytiotrophoblast, the invasive, multinucleated cell mass responsible for anchoring the embryo and initiating placenta formation. Another example is the chemotactic guidance of sperm towards the egg. The egg releases chemical attractants, like progesterone, that create a gradient, guiding the capacitated sperm towards their target. These signals don't simply tell a cell what to become. They activate specific signaling pathways, cascades of molecular interactions that transmit the signal from the cell surface to the nucleus, where it influences gene expression. Transcription factors, proteins that bind to specific DNA sequences, act as molecular switches, turning genes on or off in response to these signals. This precise regulation of gene expression is what drives the differentiation process, determining which genes are active in a particular cell type, and thus, what proteins are produced and what functions the cell can perform. The three germ layers formed during gastrulation are the starting point for this incredible diversification. The ectoderm, initially a simple sheet of cells, gives rise to not only the skin and nervous system, but also a remarkable variety of other cell types. The epidermis, the outermost layer of the skin, differentiates into keratinocytes, melanocytes, Langerhans cells, and Merkel cells, each with its specialized role in protection, pigmentation, immunity, and touch sensation. The nervous system, arising from the neural tube and neural crest, generates a vast array of neurons, the information processing cells of the brain and spinal cord, as well as glial cells, the support cells that nourish and protect neurons. These include sensory neurons, motor neurons, interneurons, astrocytes, oligodendrocytes, and microglia, each with its distinct function in sensory perception, motor control, information processing, and immune defense within the nervous system. The mesoderm, the middle layer, generates an equally diverse array of cell types. From the somites, segmented blocks of mesoderm along the embryo's back, arise skeletal muscle cells, the powerful fibers that enable movement. Osteoblasts, the bone-forming cells that create the skeletal framework, and chondrocytes, the cartilage-forming cells that provide cushioning and flexibility at joints. The mesoderm also gives rise to blood cells, including erythrocytes, the oxygen-carrying red blood cells, leukocytes, the infection-fighting white blood cells, and thrombocytes, the platelets involved in blood clotting. The circulatory system, also derived from mesoderm, includes endothelial cells, lining the blood vessels, and smooth muscle cells, regulating blood flow. The endoderm, the innermost layer, forms the lining of the digestive tract, differentiating into a variety of epithelial cells that perform specialized functions in digestion and absorption. These include enterocytes, absorbing nutrients in the small intestine, goblet cells, secreting mucus to protect the lining of the gut, and enteroendocrine cells, releasing hormones that regulate digestion. The endoderm also gives rise to the lining of the respiratory system, including the ciliated cells that sweep mucus and debris from the airways, and the alveolar cells that facilitate gas exchange in the lungs. Organs like the liver, pancreas, and thyroid gland, crucial for metabolism and hormone production, also arise from the endoderm, each with its diverse array of specialized cell types, such as hepatocytes in the liver, pancreatic islet cells producing insulin and glucagon, and follicular cells in the thyroid gland producing thyroid hormones. This incredible diversity arises from a process of specialization, a narrowing of potential. The early cells of the embryo are pluripotent, meaning they have the potential to become any cell type in the body. Even more remarkably, the very first cells, up to the 8-cell stage, are totipotent, capable of giving rise to not only the embryo but also the extraembryonic tissues, like the placenta. This totipotency, a state of unlimited potential, is a fleeting characteristic, lost as the embryo progresses through cleavage. As cells differentiate, they lose this pluripotency, their developmental potential becoming increasingly restricted. They become committed to a particular fate, a specific lineage, acquiring the unique structures and functions needed for their role in the body. This loss of pluripotency is a necessary sacrifice, a trade-off for specialization. It's what allows for the formation of complex tissues and organs, where different cell types work together in a coordinated manner. A skin cell, specialized for protection, cannot also function as a nerve cell, transmitting electrical signals, or a muscle cell, generating force. This commitment to a specific fate is irreversible, a one-way street in the developmental landscape. Each cell fate decision represents a path not taken. Imagine a branching tree, where each branch point represents a decision, a choice between two or more possible fates. As a cell progresses down a particular branch, it becomes increasingly specialized, its potential fates narrowing with each decision. A cell committed to the ectoderm lineage can no longer become a mesoderm or endoderm cell. A cell destined to become a neuron can no longer revert to a pluripotent stem cell or differentiate into a muscle cell. This irreversible nature of cell fate decisions has profound implications for the development and organization of the body. It ensures that the different tissues and organs develop in the correct sequence and spatial arrangement. It also maintains the body's overall organization and prevents the chaos that would ensue if cells could freely switch fates. A skin cell randomly becoming a nerve cell in the middle of the epidermis would disrupt the skin's barrier function. A muscle cell transforming into a blood cell within a muscle fiber would compromise the muscle's ability to contract. This commitment to a specific fate, this path not taken, is what maintains the integrity and functionality of the body's complex systems. Cell differentiation is not an isolated process. It's intimately linked to organogenesis, the formation of organs. Specialized cells, arising from the 3 germ layers, don't simply remain scattered throughout the embryo. They organize into tissues, collections of similar cells performing a common function. These tissues, in turn, interact and assemble to form functional organs. The formation of the heart, for example, begins with the differentiation of mesoderm cells into cardiomyocytes, the contractile cells of the heart muscle. These cardiomyocytes aggregate, forming the heart tube, a primitive structure that begins to beat and pump blood early in embryonic development. Other mesoderm cells differentiate into endothelial cells, forming the lining of the heart chambers and blood vessels. The heart, a complex organ with multiple chambers, valves, and blood vessels, arises from the coordinated differentiation and organization of these specialized cell types. Similarly, the lungs arise from the differentiation of endoderm cells into the epithelial cells lining the airways and alveoli. Mesoderm cells differentiate into the smooth muscle cells that surround the airways, regulating their diameter, and the endothelial cells that form the pulmonary capillaries, enabling gas exchange. The lungs, essential for breathing, arise from the intricate interplay and assembly of these specialized tissues. Time-lapse microscopy observations of morula compaction have revealed that this process may serve as a checkpoint for embryo viability. Chromosomally abnormal blastomeres, cells with errors in their DNA, are sensed and eliminated during compaction, ensuring the survival of only the fittest cells. This highlights the embryo's remarkable ability to self-correct and maintain its developmental integrity. The journey of sperm to the egg, an epic struggle against the odds, underscores the challenges faced by these tiny cells in their quest to fertilize the egg. Their capacitation, a complex transformation preparing them for this challenge, and their navigation through the female reproductive tract, a perilous gauntlet of physical and chemical barriers, demonstrate the remarkable resilience and adaptability of these cells. The formation of the morula, with its compacted cells and emerging differentiation, exemplifies the embryo's remarkable ability to self-organize, setting the stage for the emergence of distinct cell lineages and the subsequent formation of complex tissues and organs. These examples, just glimpses into the intricate journey of embryonic development, highlight the remarkable precision and complexity of the processes that shape our existence. Chapter 5. The Symphony of Development, Orchestrating the Body's Architecture. With the germ layers established as the foundation and the initial steps of cell differentiation underway, the embryonic stage transitions into a period of remarkable structural complexity, organogenesis. This chapter delves into the intricate processes by which these simple layers are transformed into the recognizable organs and systems that define the human body. Organogenesis is not merely a construction project, but a symphony of development, a meticulously orchestrated performance where genes, cells, and tissues interact in a precisely timed and coordinated manner. The overture to this symphony begins during weeks 3 and 4, a period of rapid change and dramatic transformation. Gastrulation, the process described in the previous chapter, lays the groundwork, establishing the three primary germ layers, ectoderm, mesoderm, and endoderm, from which all organs will arise. These layers, initially simple sheets of cells, begin to fold, bend, and interact, setting the stage for the emergence of the body's basic architecture. Neurulation, the formation of the nervous system, is one of the first major events of organogenesis. The ectoderm along the embryo's back thickens to form the neural plate, a precursor to the brain and spinal cord. The edges of the neural plate fold inward, creating the neural groove, which then deepens and closes to form the neural tube. This neural tube, a hollow structure running the length of the embryo, will differentiate into the brain at the anterior end and the spinal cord along the posterior. The heart, the first functional organ to develop, also begins its formation during this period. Within the mesoderm, two endocardial tubes, precursors to the heart, form and migrate towards each other, eventually fusing to create the primitive heart tube. This simple tube, initially a single chamber, begins to beat and pump blood, even as it continues its transformation. By the end of week 4, the heart has looped and begun to partition into its 4 chambers, setting the rhythm for the developing circulatory system. By week 5, the embryo, now about 4 millimeters long, has taken on a distinctive C shape. This curvature is not accidental. It reflects the rapid growth of the developing brain and spinal cord, which are outpacing the development of other structures. The pharyngeal arches, a series of pouches in the neck region, also appear during this week. These arches, reminiscent of gill structures in fish and amphibians, will give rise to components of the face, jaw, neck, and throat, highlighting our evolutionary heritage. The arm buds, precursors to the upper limbs, become visible while the liver, pancreas, spleen, and gallbladder begin their formation within the endoderm. Week 6 sees continued rapid development. The embryo, now 8 millimeters long, begins to develop facial features. The eyes and nose start to take shape, while the leg buds, precursors to the lower limbs, emerge. The hands, initially flat paddles at the ends of the arms, begin to differentiate into fingers. Internally, the precursors of the kidneys begin to form, and the stomach starts its development within the digestive system. By week 7, the embryo, now 13 millimeters long, resembles a small tadpole or seahorse due to its prominent tail and large head. This tail, a temporary structure, will eventually regress as the lower limbs elongate. The lungs begin to form within the endoderm, while the lymphatic system, crucial for immune function, starts its development. Crucially, the primary prenatal development of the sex organs begins, marking the first steps in sexual differentiation. Week 8, the final week of the embryonic stage, sees the embryo reach a length of 20 millimeters. The external ears begin to form, and the face takes on a more human appearance. Nipples and hair follicles begin to develop, while internally, all essential organs have at least started to form. The heart, now fully partitioned into 4 chambers, beats steadily at around 170 beats per minute, and limb movements can be detected by ultrasound, a testament to the growing complexity of the nervous and muscular systems. By the end of week 8, the embryo is no longer just a collection of tissues and organs. It is a recognizable human form, poised for the next phase of its developmental journey, the fetal stage. Organogenesis is not simply a series of independent events. It's a highly interconnected process, where the development of one organ influences the development of others. The formation of the notochord, for example, a rod-like structure derived from the mesoderm, signals the overlying ectoderm to thicken and form the neural plate, initiating neurulation. The developing heart, with its pulsating flow of blood, provides oxygen and nutrients to the developing organs, fueling their growth and differentiation. The lungs, initially simple buds, branch and develop in coordination with the developing circulatory system, ensuring a functional connection for gas exchange. The digestive system, from mouth to anus, forms a continuous tube, with the liver, pancreas, and gallbladder developing as outgrowths of this primitive gut. The kidneys, developing from the mesonephric ridge, connect to the developing bladder and urethra, establishing the urinary system. The sex organs, differentiating under the influence of genetic and hormonal signals, develop in coordination with the urinary system, reflecting their shared developmental origins. Organogenesis is a period of exceptionally rapid growth and development. The embryo increases in size and complexity at an astonishing rate, transforming from a simple cluster of cells to a recognizable human form in just 8 weeks. This rapid transformation is fueled by cell division, differentiation, and the coordinated movements of tissues and organs, a testament to the power of self-organization. Underlying this remarkable symphony of development are intricate molecular mechanisms, the conductors of the orchestra. Signaling molecules, like BMPs, WNTs, FGFs, and nodal, act as chemical messengers, diffusing through the embryo and influencing the behavior of their target cells. These signals activate specific genetic pathways, triggering changes in gene expression that control cell movement, division, and differentiation. Transcription factors, the molecular switches, bind to specific DNA sequences, turning genes on or off in response to these signals, orchestrating the precise choreography of development. Organogenesis is a critical period in human development, a time of vulnerability to genetic and environmental insults. Errors during this period, disruptions in the symphony of development, can have devastating consequences, leading to a range of birth defects. Genetic mutations, chromosomal abnormalities, and exposure to teratogens, environmental agents that can interfere with development, can all disrupt the delicate balance of organogenesis, resulting in structural or functional abnormalities. The occasional imperfections in development, while sometimes tragic, can also provide valuable insights into the developmental process. The rare occurrence of a child born with a tail, for example, is not a true tail, but a fatty tumor, a reminder of the complex process of neural tube closure. Ectopic pregnancies, where the embryo implants outside the uterus, highlight the importance of the precise signaling and interactions required for successful implantation. These examples, while often associated with negative outcomes, underscore the remarkable precision and complexity of normal development. As the embryo completes its transformation into a fetus, the placenta, a remarkable organ of maternal-fetal exchange, takes center stage. The next chapter will explore the development and function of this vital organ, highlighting its crucial role in supporting fetal growth and development throughout pregnancy. The symphony of development continues, with the placenta providing the essential nourishment and support for the fetus's continued growth and maturation. Chapter 6. The Placenta. A World Within a World, the Lifeline of Life. The symphony of development, having orchestrated the intricate ballet of organogenesis, now introduces a new player, a remarkable organ of maternal-fetal exchange, the placenta. This chapter explores the development and function of this vital organ, a world within a world, highlighting its crucial role in supporting fetal growth and development throughout pregnancy. The placenta is not merely a passive conduit, but a dynamic and highly specialized organ, a lifeline that sustains the developing fetus, providing the essential nourishment and protection for its continued growth and maturation. The placenta's story begins early, during the blastocyst stage, even before implantation. Recall the blastocyst, a hollow sphere of cells with the inner cell mass nestled at one pole. The outer layer of cells, the trophoblast, is not just a protective shell. It's the precursor to the placenta, the architect of this vital organ. As the blastocyst approaches the uterine lining, the trophoblast cells begin to differentiate, specializing for their roles in implantation and placental development. The trophoblast differentiates into 2 distinct layers, the cytotrophoblast and the syncytiotrophoblast. The cytotrophoblast, the inner layer, is a proliferative layer of distinct cells, the stem cells of the placenta. It continuously replenishes the outer layer, the syncytiotrophoblast. The The syncytiotrophoblast, as its name implies, is a syncytium, a multinucleated mass of cytoplasm formed by the fusion of individual cytotrophoblast cells. This unique structure, lacking cell boundaries, allows for rapid and efficient transport of substances between the maternal and fetal circulations. The syncytiotrophoblast is the workhorse of the placenta, the primary site of nutrient and gas exchange. The syncytiotrophoblast, upon contact with the uterine lining, initiates implantation. It secretes enzymes that erode the endometrial epithelium, allowing the blastocyst to burrow into the uterine wall. As the syncytiotrophoblast invades the endometrium, it encounters maternal blood vessels. It engulfs these vessels, creating spaces within the syncytiotrophoblast called lacunae, which fill with maternal blood. These lacunae, initially small and isolated, expand and interconnect, forming a network of blood-filled spaces, the intervillous space, the central feature of the mature placenta. The cytotrophoblast cells, proliferating and extending outwards, form finger-like projections into the intervillous space. These projections, the primary chorionic villi, are the first step in establishing the fetal side of the placental circulation. The primary villi quickly become vascularized, with fetal blood vessels growing into their core. These vascularized villi, the secondary and tertiary chorionic villi, branch and extend further into the intervillous space, creating a vast surface area for exchange between the fetal and maternal circulations. The intervillous space, filled with maternal blood, is the site of exchange between the maternal and fetal circulations. Maternal blood enters the intervillous space through the spiral arteries, which have been remodeled by the invading syncytiotrophoblast to increase blood flow and reduce resistance. The maternal blood bathes the chorionic villi, where nutrient and gas exchange takes place. Oxygen and nutrients diffuse from the maternal blood across the thin membranes of the villi into the fetal capillaries. Carbon dioxide and waste products diffuse in the opposite direction, from the fetal capillaries into the maternal blood. The maternal blood, depleted of oxygen and laden with waste, exits the intervillous space through the endometrial veins. The fetal blood, enriched with oxygen and nutrients, flows through the umbilical vein back to the developing fetus. The umbilical arteries, carrying deoxygenated fetal blood and waste products, extend from the fetus to the placenta, completing the fetal side of the placental circulation. This two-way street, the uteroplacental circulation, is a remarkable feat of engineering, allowing for efficient exchange of substances without direct mixing of fetal and maternal blood. The placenta isn't just a transport hub. It's a multifaceted organ, performing a multitude of functions essential for fetal growth and survival. The placenta acts as the fetus's lungs, facilitating the exchange of oxygen and carbon dioxide. Oxygen, essential for cellular respiration and energy production, diffuses from the maternal blood across the placental membranes into the fetal blood. Carbon dioxide, a waste product of cellular metabolism, diffuses in the opposite direction, from the fetal blood to the maternal blood, where it's expelled by the mother's lungs. The placenta provides the fetus with a constant supply of nutrients, the building blocks for growth and development. Glucose, the primary energy source, is transported across the placenta from the maternal blood to the fetal blood. Amino acids, the building blocks of proteins, are actively transported across the placenta, ensuring a sufficient supply for fetal protein synthesis. Fatty acids, essential components of cell membranes and energy storage, are also transported across the placenta, providing the fetus with the lipids it needs for growth and development. The placenta also provides immunological protection, shielding the fetus from the mother's immune system. It produces immunosuppressive factors that prevent the mother's immune cells from attacking the fetus, which is genetically distinct and might otherwise be recognized as foreign tissue. The placenta also transports antibodies, protective proteins produced by the mother's immune system, across the placental membranes into the fetal blood, providing passive immunity against certain infections. IgG, the most abundant type of antibody in maternal blood, is readily transported across the placenta, providing the fetus with protection against a range of pathogens. The placenta acts as the fetus's kidneys, removing waste products from the fetal blood. Urea and creatinine, byproducts of protein metabolism, diffuse from the fetal blood across the placental membranes into the maternal blood, where they are filtered and excreted by the mother's kidneys. Bilirubin, a breakdown product of hemoglobin, is also transported across the placenta for excretion by the mother. The placenta is a major endocrine organ, producing a variety of hormones essential for maintaining pregnancy and promoting fetal growth. Human chorionic gonadotropin, HCG, the hormone detected in pregnancy tests, is produced by the syncytial trophoblast. HCG signals the corpus luteum in the ovary to continue producing progesterone, which is essential for maintaining the uterine lining and preventing menstruation. Progesterone, also produced by the placenta itself, plays a crucial role in suppressing uterine contractions, preventing premature labor, and promoting the development of the mammary glands in preparation for breastfeeding. Estrogen, another hormone produced by the placenta, promotes uterine growth and blood flow to the placenta. Human placental lactogen, HPL, also known as human chorionic somatomammotropin, is a hormone that promotes fetal growth by increasing maternal blood glucose levels and stimulating insulin production. The placenta's development and function are supported by several extraembryonic structures, membranes that lie outside the embryo proper. The amnion, a fluid-filled sac, surrounds and protects the developing embryo, providing a cushioning environment and preventing dehydration. The yolk sac, while not a significant source of nutrition in humans, plays a vital role in early blood cell formation and nutrient transfer before the placenta is fully functional. The allantois, a small outpocketing of the yolk sac, contributes to the formation of the umbilical cord, the lifeline connecting the fetus to the placenta. The chorion, the outermost membrane, surrounds the amnion, yolk sac, and allantois, and forms the fetal part of the placenta, contributing to the chorionic villi and the placental membranes. The placenta, despite its remarkable resilience, is not immune to dysfunction. Placenta previa, a condition where the placenta partially or completely covers the cervix, can cause bleeding during pregnancy and obstruct delivery. Preeclampsia, a condition characterized by high blood pressure and protein in the urine, can restrict fetal growth and lead to premature delivery. Placental abruption, where the placenta separates from the uterine wall, can deprive the fetus of oxygen and nutrients, potentially leading to fetal distress or stillbirth. These complications, while relatively rare, highlight the critical importance of the placenta for a healthy pregnancy. The placenta is a marvel of biological engineering, a complex network of blood vessels, membranes, and specialized cells. Imagine a vast, spongy organ, roughly disc-shaped, with the umbilical cord attached at its center. The fetal side of the placenta, smooth and covered by the amnion, is connected to the fetus by the umbilical cord. The maternal side, rough and textured, is attached to the uterine wall. Within the placenta, the chorionic villi, branching and extending into the intervillous space, create a vast surface area for exchange. This intricate structure, a world within a world, is a testament to the power of evolution and the remarkable adaptability of life. The placenta's influence extends beyond pregnancy. It shapes the developing fetus, influencing its growth, development, and even its susceptibility to certain diseases in later life. It prepares the mother's body for childbirth and breastfeeding, orchestrating a cascade of hormonal changes that facilitate these processes. The placenta, a temporary organ with a lasting impact, is a true marvel of nature, a world within a world that sustains the miracle of life. Chapter 7. The Unseen Hand, Genetic and Environmental Influences on Destiny. The human journey, from the moment of conception to the emergence of a fully formed infant, is a remarkable testament to the intricate interplay between our genetic blueprint and the world around us. While our genes provide the fundamental instructions, the environment acts as a sculptor, shaping and refining the final form. This chapter delves into this delicate dance between nature and nurture, exploring how these unseen forces can have profound and lasting effects on an individual's traits and health during the critical period of embryonic development. Our genetic inheritance, a mosaic of contributions from both parents, determines a vast array of characteristics, from the obvious, like eye color and hair texture, to the more subtle, like predispositions to certain diseases. The 23 chromosomes we receive from each parent, carrying thousands of genes, encode the proteins that build and operate our bodies. But genes are not static entities, simply dictating outcomes. They are dynamic players, their expression influenced by a myriad of environmental cues. Think of genes as instruments in an orchestra, their individual melodies blending to create the symphony of life. The environment, then, is the conductor, modulating the tempo, volume, and even the instruments themselves, shaping the final composition. One striking example of this interplay is the role of folate in neural tube development. Folate, a B vitamin, is crucial for the synthesis of DNA and RNA, the building blocks of life. During the early weeks of embryonic development, the neural tube, a precursor to the brain and spinal cord, forms from a flat sheet of cells. This process, called neurulation, requires rapid cell division and differentiation, processes heavily reliant on folate. A deficiency in folate can disrupt neurulation, leading to neural tube defects, such as spina bifida, where the spinal cord doesn't close completely, or anencephaly, where a major portion of the brain, skull, and scalp failed to develop. The critical period for neural tube closure is between the 3rd and 4th weeks of pregnancy, highlighting the importance of adequate folate intake during this crucial window. Alcohol, a seemingly innocuous substance, can have devastating consequences for the developing embryo. When a pregnant woman consumes alcohol, it readily crosses the placenta, entering the fetal bloodstream. The developing fetus lacks the enzymes to metabolize alcohol efficiently, leading to higher blood alcohol concentrations and longer exposure times compared to the mother. Alcohol disrupts cellular processes, interfering with cell division, migration, and differentiation. It can also damage DNA, potentially leading to mutations and birth defects. Fetal alcohol spectrum disorder, FASD, a range of physical, cognitive, and behavioral disabilities, is a tragic consequence of prenatal alcohol exposure. The severity of FASD varies depending on the amount and timing of alcohol consumption during pregnancy, with the most severe effects occurring during the first trimester, when organogenesis, the formation of organs, is at its peak. Maternal infections, often mild or even asymptomatic in the mother, can pose serious threats to the developing embryo. Rubella, also known as German measles, is a viral infection that can cause severe birth defects, including deafness, blindness, heart defects, and intellectual disability, if contracted during pregnancy. Cytomegalovirus, CMV, another common virus, can cause hearing loss, vision problems, and developmental delays in infants infected in utero. Toxoplasmosis, a parasitic infection often transmitted through contact with cat feces or undercooked meat, can cause brain damage, seizures, and blindness in infected infants. These infections highlight the importance of preventative measures, such as vaccinations and hygiene practices, during pregnancy. Nutritional deficiencies beyond folate can also have significant impacts on embryonic development. Iodine, essential for thyroid hormone production, is crucial for brain development. A deficiency in iodine during pregnancy can lead to cretinism, a condition characterized by intellectual disability, stunted growth, and deafness. Iron, essential for red blood cell production and oxygen transport, is crucial for fetal growth. Iron deficiency anemia during pregnancy can restrict fetal growth and increase the risk of premature birth. Vitamin A, important for cell growth and differentiation, can be teratogenic, causing birth defects if consumed in excessive amounts during pregnancy. This underscores the importance of a balanced and nutritious diet during pregnancy, avoiding both deficiencies and excesses. Exposure to environmental toxins, often unseen and insidious, can have long-lasting consequences for the developing embryo. Lead, a heavy metal found in paint, pipes, and contaminated soil, can cross the placenta and interfere with brain development, leading to cognitive impairments and behavioral problems. Mercury, another heavy metal found in some fish, can also damage the developing nervous system, causing developmental delays and intellectual disability. Pesticides, chemicals used in agriculture, can disrupt hormone function and increase the risk of birth defects. These examples highlight the importance of minimizing exposure to environmental toxins during pregnancy. Maternal stress, often an unavoidable part of life, can also influence fetal development. When a pregnant woman experiences stress, her body releases hormones, like cortisol, that can cross the placenta and affect the developing fetus. Chronic stress during pregnancy has been linked to an increased risk of premature birth, low birth weight, and developmental problems. These effects are not simply due to the direct action of stress hormones on the fetus. Stress can also alter maternal behavior, such as smoking or poor nutrition, which can further impact fetal development. Genetic predispositions to certain diseases can interact with environmental factors to produce a wide range of outcomes. For example, individuals with a genetic predisposition to asthma may be more likely to develop the condition if exposed to environmental triggers, like allergens or air pollution. Similarly, individuals with a genetic predisposition to type 2 diabetes may be more likely to develop the disease if they are obese or have an unhealthy lifestyle. These examples illustrate how genes and environment can work together, either exacerbating or mitigating disease risk. The concept of a critical period in development is crucial for understanding the impact of environmental factors. Different organs and systems develop at different times during pregnancy, and the timing of exposure to an environmental insult can determine its effect. The critical period for neural tube closure, as mentioned earlier, is during the first trimester. Exposure to alcohol during this period is most likely to cause FASD. Later in pregnancy, alcohol exposure may have less severe effects on physical development but can still impact brain development and cognitive function. Genetic mutations, while often associated with negative outcomes, can interact with environmental factors in complex ways. A mutation in a gene involved in DNA repair, for example, may increase an individual's susceptibility to cancer if they are exposed to environmental carcinogens. However, the same mutation may have no effect if the individual is not exposed to these carcinogens. This illustrates how genetic mutations and environmental factors can interact to produce a wide range of outcomes. The intricate dance between our genes and the environment is a constant throughout our lives, but it's particularly crucial during the embryonic period, when the foundations of our physical and mental being are laid. The examples presented here, from the role of folate in neural tube development to the impact of maternal stress, highlight the delicate balance and complex interplay between nature and nurture. Understanding this intricate dance is not just a matter of scientific curiosity. It's essential for promoting healthy pregnancies, preventing birth defects, and ensuring the well-being of future generations. Chapter 8. Pushing the Boundaries: The Ethics of Embryonic Research and the Future of Human Development. The intricate journey of human embryonic development, from the fusion of gametes to the formation of a fully functional placenta, is a testament to the remarkable power of self-organization. But this intricate dance of cells and signals is not just a biological marvel. It's a source of profound ethical questions, a frontier where scientific progress and moral considerations intersect. This chapter explores the ethical dilemmas surrounding embryonic research, delving into the varying perspectives on when life begins, the potential benefits and risks of this research, and the importance of ongoing ethical discussions and public engagement. The central ethical dilemma in embryonic research revolves around the moral status of the embryo. When does human life begin? Is it at the moment of conception, with the fusion of sperm and egg, the creation of a unique genetic entity? Or does it begin at a later stage, with the development of the primitive streak, the first visible sign of the body's axis and the point at which twinning is no longer possible? Or perhaps it's the onset of brain activity, the emergence of consciousness and sentience? These are not merely scientific questions, they are deeply philosophical and moral questions, with no easy answers. Those who believe that life begins at conception argue that the embryo, from the moment of fertilization, possesses the full moral status of a human being and therefore should be afforded the same rights and protections. This perspective often stems from religious or philosophical beliefs about the sanctity of life and the inherent dignity of the human person, regardless of developmental stage. The destruction of embryos for research, from this viewpoint, is morally equivalent to taking a human life. Others argue that the embryo's moral status increases gradually as it develops. They point to the early stages of development, where the embryo is a simple cluster of cells, lacking the complex organization and functions of a fully developed human being. The development of the primitive streak, around day 14, is often cited as a significant milestone, marking the beginning of individuation, the point at which the embryo can no longer split into twins. Some argue that the onset of brain activity, much later in development, is the true beginning of human life, as it marks the emergence of consciousness and the capacity for experience. From these perspectives, research on early embryos, before the development of the primitive streak or brain activity, is ethically permissible, as the embryo is not yet considered a full human being. The 14-day rule, a widely adopted guideline in many countries, reflects this gradualist perspective. Established in the 1970s, when scientists first began culturing human embryos in vitro, this rule prohibits the culture of human embryos beyond 14 days post-fertilization. The rationale behind the rule was twofold: to avoid the ethical quagmire of experimenting on embryos with a developed nervous system and to provide a clear and practical limit for research, aligning with the then current understanding of embryonic development. However, recent advances in stem cell technology and embryo culture techniques have challenged the 14-day rule. Scientists can now create embryo models, derived from stem cells, that mimic some aspects of early human development, potentially bypassing some of the ethical concerns related to research on donated embryos. These models, such as blastoids, which resemble blastocysts, can be used to study implantation, a critical stage of development that is normally hidden within the uterus. Other models can mimic gastrulation, the process by which the three primary germ layers are formed, or even the early stages of organogenesis. These models provide a powerful tool for investigating the causes of birth defects, testing the safety of drugs during pregnancy, and developing new therapies for developmental disorders. The potential benefits of embryonic research extend beyond the development of embryo models. Embryonic stem cells, derived from the inner cell mass of the blastocyst, are pluripotent, meaning they can differentiate into any cell type in the body. This unique characteristic makes them a valuable resource for regenerative medicine, a field focused on repairing or replacing damaged tissues and organs. Embryonic stem cells could potentially be used to generate tissues for transplantation, treating a wide range of diseases, from Parkinson's disease and spinal cord injuries to diabetes and heart failure. Imagine a future where damaged heart tissue could be replaced with healthy, functional cardiomyocytes derived from embryonic stem cells, or where a severed spinal cord could be repaired with new neurons and glial cells. But the path to regenerative medicine is not without its challenges. One major hurdle is immune rejection. Transplanted tissues, derived from embryonic stem cells, would be recognized as foreign by the recipient's immune system, triggering an immune response that could destroy the transplant. Scientists are exploring various strategies to overcome this challenge, including genetic engineering to make the transplanted cells less immunogenic and the development of immunosuppressive drugs to dampen the immune response. Another challenge is the ethical concern related to the destruction of embryos to derive embryonic stem cells. While some argue that the potential benefits of regenerative medicine outweigh the moral concerns, others remain steadfast in their belief that the embryo deserves full moral consideration. This ethical dilemma underscores the need for ongoing discussions and public engagement. Embryo models, derived from stem cells, offer a potential solution to this ethical dilemma. These models, while not identical to human embryos, can mimic many aspects of early development, potentially reducing the need for research on donated embryos. Blastoids, for example, can be used to study implantation and the early stages of placental development, providing insights into the causes of early pregnancy loss. Gastruloids, models that mimic gastrulation, can be used to investigate the formation of the three primary germ layers and the early stages of organogenesis, providing clues to the origins of birth defects. The development of these embryo models is a rapidly evolving field, with new and more sophisticated models emerging constantly. These models hold immense promise for advancing our understanding of human development and for developing new therapies for a wide range of diseases. But they also raise new ethical questions. How closely do these models need to resemble human embryos before they are subject to the same ethical considerations? At what point does a model become an embryo? These are complex questions that require careful consideration and open discussion. The ethical considerations surrounding embryonic research are not static. As science advances and our understanding of human development deepens, the ethical landscape shifts, requiring ongoing dialogue and reevaluation. The 14-day rule, once considered a firm ethical boundary, is now being debated in light of new scientific possibilities. The development of embryo models raises new ethical questions about the moral status of these entities. These ongoing discussions are essential for ensuring that embryonic research is conducted responsibly and ethically, balancing the pursuit of knowledge and the potential for medical breakthroughs with respect for human life and dignity. Public engagement in these ethical discussions is crucial. Scientists, ethicists, policymakers, and the public must work together to navigate this complex terrain, ensuring that embryonic research aligns with societal values and serves the common good. The human embryo, a microcosm of our potential, holds the key to understanding our origins, our health, and our future. By engaging in open and thoughtful discussions about the ethical implications of embryonic research, we can ensure that this powerful tool is used wisely and ethically, unlocking its potential for good while upholding the fundamental values that define us as human beings. From a single cell to a complex being, human embryonic development is a breathtaking journey of intricate processes. The fusion of sperm and egg, a seemingly simple act, initiates a cascade of precisely orchestrated events: cleavage, blastocyst formation, and the critical process of implantation, where the embryo establishes its connection with the mother. Gastrulation, a period of dramatic cellular rearrangement, lays the foundation for the body plan, forming the three primary germ layers: ectoderm, mesoderm, and endoderm, from which all tissues and organs arise. Cell differentiation, driven by a complex interplay of genetic and environmental signals, transforms these generic cells into the specialized components of our bodies, from skin cells and neurons to muscle cells and blood cells. Organogenesis, the formation of organs, builds upon this foundation, assembling these specialized cells into functional units, from the beating heart to the developing lungs. The placenta, a remarkable organ of maternal-fetal exchange, emerges as a lifeline, providing nourishment, oxygen, and immunological protection throughout pregnancy. These intricate developmental processes are not solely determined by our genes. Environmental factors, including maternal nutrition, exposure to toxins, and even maternal stress, can have profound and lasting effects on the developing embryo, highlighting the delicate interplay between nature and nurture. Understanding these influences is crucial for promoting healthy pregnancies and preventing birth defects. The remarkable progress in understanding embryonic development has opened new avenues for research, including the development of embryo models derived from stem cells. These models offer powerful tools for studying early human development, potentially bypassing some of the ethical concerns associated with research on donated embryos. They hold immense promise for understanding the causes of birth defects, testing the safety of drugs during pregnancy, and developing new therapies for a wide range of diseases. These advancements, however, raise profound ethical questions about the moral status of the embryo and the limits of scientific intervention. Ongoing dialogue and public engagement are essential for navigating these complex issues and ensuring that embryonic research is conducted responsibly and ethically. The future of human development research hinges on our ability to balance the pursuit of knowledge and the potential for medical breakthroughs with respect for human life and dignity. This exploration of human embryonic development has revealed the intricate dance of life, the remarkable journey from a single cell to a complex human being. Where do you see this research leading us? Share your thoughts and questions in the comments below. Let's continue this conversation together. Don't forget to like, subscribe, and suggest topics for future videos. The journey of discovery never ends, and the pursuit of knowledge is a path worth exploring.